Well, today it is my enormous pleasure to be talking once again to Edward J, or Ted to his friends, and hopefully I'm a friend now, Ted Steele. Ted, I've spoken to Ted on several occasions previously, a couple on my own platform here on Empower Ed. Ted is a molecular immunologist, and we'll get to what that is in a minute, with interest in virology and evolution. And Ted and I were just emailing back and forth about something recently, and I asked Ted if there was anything of interest that he had that he would like to discuss, and he said, "Well, I got this paper, which is essentially a summary of his history as a scientist developing ideas of Lamarckian inheritance." And I said, "Of course, I want to talk about that." So, Ted, it's so nice to have you back on, and let's talk about this fellow Lamarck and about evolution and all that jazz. So could we maybe start, assuming that my audience isn't as up to speed as you are on evolutionary theory? How about we start with just a little potted history on the development of evolutionary theory? So, Darwin, Lamarck, the ideas that both of them had. Let's go with that.
Okay, well, let's give a historical chronology timeline so it's clear. In 1809, Lamarck published his Zoological Philosophy, a major book on evolution, and it was basically, his ideas on the way life transformed from one species to another, how life might have started. But so they're a bit early, his ideas, but one core assumption, Robin, in his theory was the idea that acquired characters are inherited. Now, that wasn't controversial back then. That was a commonly held belief among many observers of biological species and biological phenomena.
Including Darwin himself, as I understand it.
Yes, that's right. Yeah, well, before Lamarck published in 1809, Erasmus Darwin, Charles Darwin's grandfather, was talking about talking about the inheritance of acquired characters. In 1859, when Darwin published his Origin of Species, if you read that book carefully, it's actually littered throughout the book with Lamarckian explanations. That is, all sorts of phenomena that could only be explained by of use and disuse, which could only be explained by the parents exercising their faculties and organs of their body, before they and their offspring inherited some of that acquired learning during life, whether it be feeding or defensive actions or flight from predators and so forth.
Sorry to interrupt. I wanted to underline that use and disuse side of it, because that's very, very pivotal. In other words, behaviors that creatures engaged in, and
yes.
Let's just say strategies that they used that made them more successful, could be passed on to their offspring.
That's right.
And not just in a sort of transmission of culture, the way that adult animals will teach their offspring.
But aspects of that culture, if they involve, metabolic features, physiology, structure, that could get embedded in, in the inheritance as well, the acquired inheritance. So they were intimate, but it wasn't controversial. That's the first point. And when Darwin published his Origin of Species, he actually made, unfortunately, some snide remarks about that fellow Lamarck. But then he used all of his same explanations and arguments in his explaining things. That's typical among scientists. They can often do that.
Right.
And that's so. But you see, in 1868, Darwin himself, from a lot of his plant breeding experiments and grafting experiments, putting a cutting on a different stem that's rooted to the ground, and looking to see whether some of the new seeds that are produced by the cutting are taking on any of the characteristics of the stem, and he found that. But that's a very well-known phenomenon in plant grafting and plant hybridization and plant breeding.
Totally. I've got a bunch of grafted fruit trees.
Well documented. Oh yeah, well documented. well documented. Anyway, the point was he came up with a theory. See, I'm going to try and stick to principles and concepts mainly in my talk with you. His theory was that the uh, pan genes enwrapped. He had no idea what genes were. He had no idea, but he coined the term pan genes or gemmules, which were transferring information in the case of his plant hybridization experiments from the stem to the graft, which produced where the seeds were produced, and that so things were in motion. That was the important thing. Genes are in motion. He didn't know what genes were. This was well before Mendel's work hadn't really been discovered properly or documented, so that was still forming too. But so you've got to understand, Darwin himself was an outright Lamarckian, and that's some of his most important contributions were there, as well as natural selection principle. That is, those that survive. It's actually a tautology, really. Those that are the fittest in that environment survive and produce children. Well, that's fair enough. That's an obviously logical thing that can happen. But in 1883, August Weismann, a German scientist and biologist, was affronted by Darwin's pangenesis theory. So he actually produced a string of different experiments. His main one, the tail chopping experiments in mice, to show that lopping off the tail of a mouse never resulted in a mouse producing offspring without tails. Of course, these are all part of the deep history of the battles, but everyone believed that was very convincing for a lot of people for some crazy reason. And I say it's crazy.
That's not a use-disuse case, is it? It's not a case of the mouse stopped using his tail because he didn't need it anymore or something like that, or he discovered a better way of locomotion. God only knows. Someone chopped off his tail, so I can't see how that's.
You've just touched on what they should have been looking for. Altered behaviour in the mouse offspring that didn't have a tail. That's what they should have been looking for.
How did they?
Yes. Anyway, it had a powerful grip on the human mind, like many simple denial. So the Weismann barrier came into existence, which fundamentally denied any somatic modification in the body, through adaptation, active adaptation, and we have to say before reproductive maturity. We've got to make that very clear, because that's a logic gate in understanding all the later data that I had to deal with. People had forgotten that. But anyway, by the time of the turn of the century, the Weismann barrier was firmly in place, and then there were, in the first 20 years of the 19th century, the 20th century, there were some really highly virulent debates going on about the inheritance of acquired characters, and the penetration of the Weismann barrier. And these were experiments by Paul Kammerer, a Austrian biologist and zoologist, and he was looking at the mating behaviour of the midwife toad and other salamanders. He was actually looking at regrowth and regeneration of limbs and all that sort of stuff. And anyway, that was just as the First World War was ending. His experiments, which he did prior to the First World War and in the war and in the intervening period before the war, his institute, he was on the wrong side of the war, of course. So his institute got destroyed. A lot of his evidence was, and his samples, some of them survived, but then the critics of his work on the penetration of the Weismann barrier did their utmost to demonize and smear him. And I say that because it took another 50 years or 40 years or so for Arthur Koestler to actually examine the whole, it was like the Dreyfus case in France, the whole hysteria surrounding Paul Kammerer. And many of the experiments now are all understandable in terms of some of the epigenetic sort of concepts we have now in Lamarckian inheritance. And so it looked like malicious individuals were touching up graphs and touching up things deliberately to discredit him.
Yeah, gotcha. I do want to come back to the crossover between what is now known as epigenetics and Lamarckian
exactly.
inheritance.
That's very important. And we must get to that.
Yes, yeah, that's exactly what I think. It's just crucial. Do you think there was there, aside from just the pride of scientists
Yeah,
and the
yeah,
determination of scientists to nail their colours to the mast, do you think there was any kind of socio-political undertones to this? I mean,
yeah. I
I'm just
suspect so, although I'm not an expert on that. But certainly, if you read um, Arthur Koestler's book, The Case of the Midwife Toad, he documents all the currents and undercurrents and political intrigues going on during that period. And of course,
gotcha.
and he did that because he actually believed that Camera was basically right, and that's what I did. So Arthur Koestler himself was a lightning rod for political controversy. He was a major figure in the prior to and after, and the fall of communism, and he was a major warrior, if you like, in the political debate, as well as a brilliant writer and a brilliant historian. He wrote some very important books on the history of science. People forget. Anyway, it's now in a more cooler light. A lot of Camera's experiments now are understandable, and other experts have crawled over them. We cite them in our papers, and point out that they're quite reasonable now in the light of current modern knowledge. But really, the biggest one that demonised Lamarck, we've got to get this very clear, was the rise of communism in Soviet Russia, and particularly the rise of Joseph Stalin, and the whole terror, of the Soviet regime, and the whole need to prove that that human beings were malleable and could be conditioned and changed by the political ideologies of the time. Okay, I can't hear you, Robin.
I muted myself because my dog was shaking. Yeah. So, and this is why I asked you before whether you thought that there might be some sort of socio-political element to
Oh
this
yes,
pressure
and it was really, yeah,
ideas.
the appointment. Sorry, go
Yeah,
on.
because eugenics was so central to the
Yes, that was
ideology
also.
of, yeah.
Yeah, that was
Reform.
another current I haven't even mentioned. Yeah, that was also going on as well. Uh,
Yeah.
but but the biggest, most damaging one to the whole concept of even taking Lamarck's ideas, or the summary of that whole period seriously, was Lysenko. He was a biological scientist and a plant breeder that Stalin appointed as his head of agriculture, and to prove to the world that they could grow wheat in Siberia, and therefore they could adapt wheat. And there was some some early signs of some success in some of these experiments. But the point was, it was a highly political KGB operation, in the sense that anyone that opposed Lysenko was immediately sent to the Gulag and
Yeah.
lost their life in many cases. So that was totally beyond the pale for Western scientists to even,
Sure.
even I felt the same way. You see, so you can imagine that force coming in. So look, when I entered the scene, because really we're setting the stage of why would a young Australian scientist in the 1970s enter the scene? Well, first of all, I was inspired by Arthur Koestler's documentation of many of these things in his books, but also too, there were logical reasons to give Lamarck a bit of breathing space in the mid to late 1970s, when I entered the scene. But the suppression against those ideas was overwhelming. Look, you know, I couldn't do it now, but as a young man, I thought I could do it. I thought that I could take on these forces and survive. So I've just got it in a summary. In summary, the atmosphere, the zeitgeist of biology then was, it was forbidden for any biologist to talk about Lamarck or take it seriously. Okay,
Mm.
so when I started talking about it, of course, I was immediately identified as an heretic.
Yes.
I just got
I
my
can well
parents' death.
imagine. And again, for people who really don't have any knowledge of this field, I think it's impossible to sort of to overestimate just how seriously this kind of Lamarck versus Darwin
Oh,
divide was laid. I mean, I can
mm,
remember studying at a fairly basic level, molecular genetics molecular biology at UNE. And it was,
mm.
oh, Mark, what nonsense, what piffle, what tosh. We're
Yes,
all
yes,
we're all
yes.
Darwinists over here. And that was in the late 90s, early 2000s.
Oh yes, it's still going on, by the way. And like, for example, because of my involvement in these sorts of controversies, I've been, like many other people, I join them pretty respectful. I've been banned for life from Wikipedia. They completely allow distortions about me and my work to get published, even down to the molecular level. The DNA sequence level for somatic hypermutation. They won't even allow me to correct obvious errors or over. Yeah, and it's extraordinary.
I was
Anyway,
actually
that's a
surprised.
side issue, but it's an
I was,
indication of the extent that this goes
yeah,
to. That's all.
yeah. No, I was surprised, Ted, at how relatively non-hostile your Wikipedia profile was. Because I went to it expecting, oh, this is just going to be a total drubbing of Ted. It's just going to be Ted's a conspiracy
No, it's a
theorist and Ted.
distortion. It's actually a distortion.
Yeah.
There's errors in it, and I can't correct them.
Right, right. So it's not a total panning of you, but they say things that aren't true and then disallow you from correcting it. Yeah, that's the Wikipedia game.
I just can't correct the obvious, but it's right down to the nucleotide level, the DNA sequence level for the mechanism of somatic hypermutation. That's what they won't allow me to correct. See, that is now a flashpoint. The reverse transcriptase model of somatic hypermutation is a hangover of the same debate, the Darwin versus Lamarck debate. Anyway, look, we've deviated a little bit.
I'm going to need you to explain that part. Is that the right time? Is it the right time to talk about reverse transcriptase?
Well, yes. Well, look, maybe I should, and I'll do it. I'll put it in context. I'll give you the context of what the intellectual and scientific zeitgeist was in the 1970s. It was firmly against many of these points I'm going now, but I should say this. The weight of evidence since the 1970s now literally favours Lamarck. Lamarckian inheritance in all sorts of ways. And there are about 4 or 5 levels of where it favours it. First of all, there are numerous examples now, not just from the early experiments I did with Reg Guzynski, but numerous examples of other laboratories showing transgenerational acquired inheritance. That is, you expose a male or a female to an acquired adaptation, whether it be, and in the biomedical field, it would have been in things like diabetes, acquired diabetes. And then you look for some of these symptoms propping up in the progeny without the original treatment that caused the acquired effect in the first place being applied. But that's, and I'll get to that if we want to. But the point is, transgenerational, phenomena were forbidden by the Weissman barrier. So how was that possible? That's the first thing. The second thing was because that implied the penetration of the Weissman barrier in some form. Then, of course, we had in the 70s Howard Timmon, and then confirmed by David Baltimore, had discovered the enzyme reverse transcriptase, which copies RNA back to DNA. And that's very important because my first theory, the somatic selection hypothesis on how Lamarckian inheritance could be affected, actually depended on that process. And that was built into, so a reverse transcriptase, if you like, I'm going to use a technical term, a reverse transcriptase, it's an enzyme, linked or coupled soma to germline mechanism was the core of that 1979 theory. So let me just, in more general terms, this is how the immune system could adapt its antigen binding recognition repertoire to the parents providing some sort of feedback, setting up the next generation for using some of that information that they used in fighting off a pathogen for their children. Now, there are roadblocks to immunologists grasping that, but they haven't fully understood what that means. But anyway, that's why we had to write the whole history again and all the evidence. But my major concern was at 2 levels, was doing transgenerational experiments or looking at the evidence for them. This is from the late 1970s. And then also a major research program on understanding how the immune system itself used reverse transcription to adapt to disease and fight off unexpected infections. And that process is called somatic hypermutation. So I've been working in that field for a long time, 40 odd more years, and advanced about 40 years ago the reverse transcription mechanism as a hypothesis, but based on evidence. So by the way, I don't put hypotheses just out of thin air. I anchor them in very solid evidence, and so I know the foundation of what I'm saying is solid. Okay, and that the reverse transcription model now is basically confirmed at many levels of molecular immunology. And I don't want to go into the
Again,
details.
I'm just going to interject here again for the listener who is not as
Yeah,
literate in genetic concepts. So
yeah,
the central dogma of genetics essentially is you've got your DNA, and then a section of DNA is read into RNA, which then instructs the protein making apparatus within
yes.
the cell to make a particular protein. So it's DNA, RNA produces a protein.
And that
And
is a dogma, and that's a legitimate dogma. That's a central dogma,
yeah,
yes.
yes. And then, so what you're saying is the discovery of reverse transcriptase provides the mechanism by which the RNA can write back into the
Yes,
DNA. So, in other words, the experience that is being undergone by that particular cell and that particular organism can, there is a mechanism by which that experience can result in the changing of a
yes,
gene.
yes. And the tip of the spear of that concept is the mechanism of somatic hypermutation, because that goes on within the body. Okay, after you vaccinate, a portion of the cells, some of them are reacting through their germline genes, but a portion of the cells mutate their germline genes that are now expressed in the white cells, and then the cell adapts to produce the best, the fittest, if you like, strongest binding antibody to rid, and that's very simple, rid the body of the pathogen. And
Yep.
the best way to understand all of that data, I won't go into it, because it's too technical, is through a reverse transcriptase model. And the last 10 years, the last 5 or 6 years, we've published a number of papers now that are getting through the peer review system, documenting all that evidence. Some of the peer review, it's often hard to get a peer reviewer, but when I get one, as the editors said, we're finding it hard for anyone to engage you. And they'd find someone, and then of course it's easy to handle the objections because the evidence supports what I'm saying. That really is another issue. It's been very, very difficult to have any sort of engagement with the field, and they've gone completely silent now. Now that the evidence is built up to, it's now so the treatment I get now is silence, basically. It's not acknowledgement. And Andrew Franklin, who doing his PhD in 2003, discovered the key enzyme has reverse transcriptase properties, that is DNA polymerase E. That's a highly technical term. But Andrew Franklin, Bob Bland, and my student made a key discovery, and that disrupted the field a little bit. But they wanted to put it to the side. They didn't want to talk about it because that really upset people, because prior to that, this particular enzyme was just thought to be, just mutating DNA, whereas this enzyme can cause RNA mutations. You see, and RNA mutations can get fed back to DNA, and then they can be part of this new set of antibodies that are being produced, fighting the disease. Now, just here though, everything we're talking about is based on Darwinian principles. This is the other problem that often people trying to talk or engage me forget. I'm not against Darwin's natural selection principles because they operate at all levels, right down to the molecular. But when it comes to somatic adaptation, the first theory that I put forward, called somatic selection theory, basically took the focus from selection in the wider population now to the selection of cells within the body in different organs adapting to different changes. And that's perfectly understandable. So that's what somatic selection means, that as the body adapts and changes, the immune system is a clear example of how that would happen. Some of that new change that gets embedded now in the DNA of the genes of the somatic cells can in fact get back to the germline. Now, that's where we've really got to talk about epigenetics and the other bit now with Lamarck, because that is a key concept. Do you want me to talk about that? Because
Please do.
Well, look. Why don't I do that? I'm just going to have on my own screen one of the from my papers. Just 1 second. Look. You probably can't see this, but I'll describe it in words. There are two phases.
Share it if you want. You can
but
actually.
it's really a figure from the bigger paper that you would have got. Now, look, I'll just describe it in terms. There are two phases to an adaptation, as we've summarised. There's an epigenetic phase, of Lamarckian inheritance. And this is demonstrated by a number of real phenomena that were human phenomena, like the Dutch famine at the end of the Second World War, the runting and the stunting behaviour in several generations, but that eventually got bred out of the population and they recovered.
Yeah,
Okay.
and sorry again, just filling in the background for people who are not familiar with this. So the Dutch hunger winter was a period of extreme caloric deprivation of the Dutch people
Yeah.
when the Nazis overran
And,
the Netherlands. You know, they took
yeah.
all their livestock, and the people were
Yes.
basically on very much starvation rations. And there were women who were already pregnant at the time
Yes.
of this famine. There were women who became pregnant during the
Yeah.
famine. So there's been a very, very long-term follow-up project to track the health
Yes.
outcomes of people who were conceived and born at various points. And
Yes.
the long story short is people whose mothers were pregnant with them while the Dutch hunger winter was on, have a higher lifetime risk of cardiovascular disease and type 2 diabetes. So
Yes. That's right.
that's really what we're talking about. Yeah, sorry.
Yeah.
Back
Yeah.
to you.
Well. Yeah, and because they eventually reversed it and bred it out, it became a symbol of reversible nature for epigenetics. Epigenetics means above the genes. It's to do with regulation of expression of genes, and it's molecular markers identify the genes that want to be expressed. So it's really about the expression of genes, and and uh, and it's the targeting process. Different tissues and organs in the body get targeted by epigenetic marks on expressing certain genes that are needed for that change. And
The analogy that's often used is if the DNA is like a big long recipe book
yeah,
with all the family recipes for 3 generations, then the epigenetic markers are like paper clips that will sort of clip out various pages and say, look, don't make these recipes, they're no good. Here, make this one over here.
that's right. Suppress these and enhance those genes.
Yep.
That's what it does. And so that's epigenetics. Now, as we've shown we've generalized this somatic hypermutation concept to somatic mutations in cancer and other things. So we've taken this concept a bit further, and it's clear that this key enzyme that Andrew discovered is a pretty accurate enzyme. It copies RNA back to DNA, but if RNA gets any mutations in it through copying an altered DNA template, for example, that is, so an altered portion of a gene that's been altered by some other process that directly changes that gene, or
Like
through,
radiation, for example.
well, the somatic hypermutation, I don't want to get into too much detail, is triggered by a direct DNA mutation process that
Okay.
triggers the process. But once it's triggered, that gives the opportunity for RNA mutations to be generated by another process that mutates RNA, and these sort of processes together can get dysregulated during the early soft epigenetic phase, and if it results in those expressed genes now mutating somehow, somatically mutating, which they could because it increases the probability of that happening, but are compatible with the cells in that tissue or organ, whether it be liver cells or pancreatic cells, whether it's compatible with that cell surviving and functioning. That the change is not lethal. Because look, if there's any lethal somatic mutation in a body cell, there's a natural process called apoptosis, or just programmed cell death. You get purification, you get elimination of the cell. Anything that
Yeah.
survives that's a benign change, or a subtly different version of the gene that's being expressed, that will survive. That's what we mean by somatic selection. You see, now if so, following the epigenetic targeting phase, which is reversible, you could transition very easily to the mutated phase, and then we've got to get the the altered somatic RNAs, particularly in these selected cells, getting back to the germline. And the various well-documented mechanisms now for moving lots of molecules, but particularly cargoes of RNA and DNA, and whether they're mutated or not, from one cell to another. And these are called vesicles or exosomes. In the original theory in 1979 that I put forward, I invoked retroviruses, that harmless endogenous retroviruses as the vehicle for the transmission of these molecules. But it's clear now that extracellular vesicle communication of various types of cargoes representative of the cell where the vesicle got emitted from. So it looks like Darwin's gemule. It really does, and people have drawn that analogy quite strikingly in certain situations. That's how packages of genetic information can be emitted from an excited cell. Now that's might have changed. Now it's getting the RNA, particularly the RNA or the DNA, but particularly the RNA that's made from this altered cell into the germlines. That is the progenitor cells for eggs and or later, because the egg is whether it's early or whether it's mature, ready to be fertilized, is a larger cell, whereas sperm, the major changes would have to go into the mother cell, the spermatogonia, because a mature sperm itself has hardly any cytoplasm or any capacity to take anything up. It can take a few things up, but not the sort of thing we're talking about with vesicles and exosomes. But anyway, so that's how we've summarised in our review all of the critical steps in getting an acquired adaptation into the germline, and we're doing that to try and explain all of these transgenerational phenomena. You see, now let me just go back. I talked about epigenetics, which is soft, because that can be reversed. But once you get the soft to hard transition on the lines I've described, that is, there's genetic changes now going on in that target organ, and some of that could get back to the germline. Once you have that, you have the opportunity for hard Lamarckian inheritance. So the major stimulus for this recent big paper we've published reviewing all this was trying to reconcile an extensive literature now on experiments on soft in mice, mainly in mice, on epigenetic inheritance. And it involves, two main thrusts. There's the maternal stressor, maternal stress and separation responses and the effect on the progeny growing up. The sort of stresses would be things like removing the pups, the newborn babies, from the mother for several hours and putting them back, but doing it in an unpredictable sequence, so the pup doesn't know where the mother is, the mother doesn't know where the pup is. And then, or putting the mother through stresses like having to swim for half an hour in a small tank, and then and cold, so if they do that to the mother, they find they can produce male offspring which are displaying all sorts of aberrant behaviour by their various tests and the various different cage arrangements. They can do highly quantitative. Say, is this male pup or progeny? Is it behaving in a normal way or not? And so that is the metric. Now, those sort of experiments,
and sorry, just to be clear, so the mother is subjected to these stressors before she becomes pregnant with the litter. Okay, so you're not testing the effects of stress.
No. It can happen after, and
right.
So that's getting back to this point of this, because the key group, Mansai in Switzerland, the key group that have done these experiments, have shown that the factors that are being transmitted to the male, that then appear in subsequent generations, by the way, are being transmitted by factors like vesicles and episomes in the milk, in the serum, the plasma of that mother. You can use that to then modify, and modify. Because it's all genetically inbred and all controlled, and you can actually do really highly controlled experiments. But it's highly contrived. But that's the way they've broken it down, and it's so aberrant development and behaviour can get transmitted down the generation. But the key thing that attracted me to all these experiments, knowing the other data that I know about, was the way they do the experiment. Because the way they do their experiment is rather intriguing. Because they want to prove that they're dealing with a Lamarckian epigenetic effect, a reversible effect, They take the affected male pup from either from the first generation, the second generation, or third generation, and mate that male to a virgin female who hasn't had, is totally normal. And then they ask, do the offspring of that affected male from the original grandmother parent who had all these things done to her, in trying to look after her babies, is that still appearing in third or fourth generation? Well, what they find in all these experiments, it can be transmitted that way by selecting out the male and mating with a virgin female, a normal female. It can be transmitted to fifth or sixth generation. So, but then it wanes and dies. So therefore, they say it's reversible. Now, that's when I came in recently. I just couldn't understand why they were doing experiments like that, because I knew of other transgeneration experiments where let's forget about the acquired adaptation first of all, but which were cropping up in the offspring. But the experimenters in that era, the 50s, 60s, and 70s, they just let the offspring. Look, the thing is, I still haven't learned how to use these mobile phones, probably, by the way, how to stop these things. But anyway, look, the point is that these experiments were highly contrived, testing it through the male. But then the other experiments in the 50s, 60s, and 70s, in both the immune system and in the metabolic system, that is acquired diabetes, the experimenters let the offspring of the different generations interbreed or even inbreed amongst themselves. And that really created stable Lamarckian stable lines of spontaneous diabetes cropping up in the fourth, fifth, sixth generation, acquired autoimmune disease cropping up in the fourth or fifth generation. So, and in another case,
if two mice whose mothers, sorry, go on.
Well, it occurred to me, just to give you the punchline. Yes, what these experimenters should be doing is probably allowing a bit of inbreeding and interbreeding to occur in that first generation, not just relying on the male to carry the whole load of transmitting the acquired effect. So it's really operational. It was a really operational prediction suggestion we've made, because those are other experiments of stable Lamarckian inheritance clearly weren't done that way. So that means that the way you breed down the generations, and then of course I ran into the whole edifice of, but that's inbreeding, that's forbidden. But of course, in our ancestors, it might have been forbidden, but it went on in small villages and small tribes.
It most certainly did. And there is I think it's a bit more than a theory. There is evidence that the human population at some point dropped really catastrophically low, and there were maybe a couple of thousand adult humans left on Earth, and so we're basically all descendants of that very low number of stock.
And in a bottleneck, and a bottleneck will increase the opportunities for that sort of inbreeding. But look, of course, other things that I say, we say, that I drafted this particular section if just surveying the human races and populations and the way the emergence of all the civilizations over the last 100,000 years. The extraordinary thing is that we can actually see distinctive genetic differences between the races. Now, I'm not being a racist when I say, I'm just being an observer. But the point is, it's very hard to talk about this, but you can see very distinctive differences between Chinese, Japanese, Caucasians, Arabs, Persians. There are so many distinctive things that you wonder that the founding populations in all these cases would have been, political and hierarchies, and probably a lot of inbreeding went on. Amongst first and second families.
Some of it was done for power reasons. I mean, the Egyptian royalty, for example, really favoured brother-sister marriages to kind of keep it in the family.
Absolutely, yes.
The same was true. I mean, endogamy is still practiced, particularly among Ashkenazi Jews, which is why there's such a very high prevalence of certain genetic disorders like Tay-Sachs and even the BRCA gene, because they're very highly endogamous.
Yes. So for me, it's really just pointing out that the natural evolutionary history of the different races of mankind. It's probably, this has been a striking feature of it. So you've got to understand, we're making these suggestions from the operational point of view. If you want to show these things definitively, then the next thing is, how does it happen? How does an incoming sequence search out the locus that it has to, that is the place in the genome where it has to insert this new sequence, this RNA sequence. And there are very respectable models and phenomena now which show that whilst conventional, as we have to understand a little bit about genetics, in the double helix, there's a conventional hydrogen bonding, between A's and T's and G's and C's. That's the conventional way that the genetic code is propagated from one generation to the next. But there is the phenomena of and John Maddock, he's the principal group that's really discussed this. There's the triplex recognition process where, say, in simple terms, it's a very important concept because it solved for me a lot of problems. Let's say a modified RNA molecule, maybe even a long one that's involved in regulating genes, gets in by a vesicle into the germline and is being, but that RNA sequence has somehow got to line up with the place in the DNA where it's got to get in and replace the resident sequence. See, true Lamarckianism is that while there might be errors in that process, that's how it would happen. It'd be like slotting in new programs into modern computers in a very selective way,
like a patch,
like a patch, yeah, like a patch insertion, yes. And so there's a thing called Hogstein base pairing, which is a completely different principle from hydrogen. It's a form of hydrogen bonding, but it's not really, and it allows recognition of long. RNA can recognize long sequences, up to hundreds, probably thousands of bases in a row, and recognize it, and then that becomes part of the targeting process in the DNA. So we've raised that as an important step. Now, we don't go into further molecular detail. I just needed to know whether conceptually it was possible to do that, and of course, John Maddock's work shows that it's possible to do that.
Yep.
So you can see how we've written this review of all the key evidence over the last 50 years that favours these processes. Now, of course, the other thing is that DNA, that enzyme that our student Andrew Franklin, discovered was a reverse transcriptase, that has very special integration properties. It actually allows the incoming RNA to be inserted right into the spot you need it to be inserted to. So you see why I view the evidence in a very a hierarchical way of key steps, and all of that is in place now, and that leads us to some of the other things that we had to talk about. Anyway, look, maybe I'll better let you ask your question. Just to sum up, Lamarckian inheritance is a two-stage process: soft inheritance, hard inheritance, and there are highly legitimate ways of thinking how that transition can happen now at the molecular level and at the cellular communication level. The emission of a vesicle-loaded cargo or RNA molecules from one part of the body and could get into another part and then into the germline.
Yeah. So to sum up, we have evidence that Lamarckian inheritance, that is the inherited, that is the transmission, let's say, of behaviors that. And these could be both behaviors as invisible things that the creature does, and also metabolic adaptations that enhance the chances of survival. That these can be passed on. Not in the and sorry, actually, what I wanted to do is to contrast Lamarckian inheritance with the idea of evolution through natural selection, and if this is a too crude of an oversimplification, please feel free to jump in and correct me. But as I understand it, so natural selection basically occurs through random mutation, where mutations in DNA just randomly occur, and most of them are not conducive to the survival of the species, but some of them are. And so the ones that are will persist, and then the creature that has these particular mutations will be more fit for its particular environment, will be more cold tolerant or heat tolerant, and will be able to eat this food that the other members of that species can't eat, and that's the survival advantage, or whatever the heck it is, right? And so this will then persist in its offspring. However, any adaptations that the creature makes during its lifetime cannot be passed on to its offspring. Yeah, because of this Weissman barrier. Right. Okay.
So it's purely random selection, random mutation and selection, just by chance that the offspring from that parent will have the change not necessary for the survival. Yes, it's random. That's the key point.
Yeah.
Yes.
Yep. And so the contrast between that and Lamarckian selection is that Lamarckian selection allows for a much more tailored process of adaptation of the species to its environment as that environment is changing. Yeah,
that's right.
So it's much more rapid, and that's so one of the criticisms made by for example James Tour is that, and his understanding as a molecular chemist is that Darwinian evolutionary processes are just too slow to explain.
Too slow.
Yep. So you would agree with James Tour on that?
Oh, absolutely, yeah. But he's not the only one that's been saying that. People have been saying that for years in all sorts of ways. And the point is that if you just look at the rise of mankind and the evolution, it's been extraordinarily fast. Let's just take from 2 million years ago when the emergence from the great apes, but just take the last 100,000 years where all of the many major changes have taken place. It's really hard to imagine them occurring by chance. You see, the whole point about the Lamarckian process is that provided all happens before the parents who survive get to reproductive maturity, and it can be both male or female. It's not restricted to the males, although there are good reasons to think that the males would, because of the constant production of new sperm throughout life, can be important. That provided that happens before the change happens before they produce their offspring. That sets up the offspring to be prepared for some of the likely changes and new environments that they're going to confront. You see, that's why it's important, and therefore it reduces the odds substantially. But it's now got a genetic base to it. It's not just random anymore, but the new genes, the new substratum that offspring is now dealing with, is setting it up to handle that environment better. The immune system is a classic example of that. But that's the key thing. Random mutation in germline genes versus updated, directed mutation, if you like, by Lamarckian inheritance and relevant to the likely environment that the offspring are going to be in.
Yep. There are so many directions. There are so many questions I want to ask. But let's I do want to discuss this issue of the immune system and its function, because we have these numerous examples where European colonists, settlers, invaders, whatever you want to call them, I'm sure the context was different in each environment, they had encounters with some native people who had not had exposure to measles or smallpox or typhoid fever or whatever the heck was rife in Europe or the colonizing countries at that time. And what happened in all of these encounters between Indigenous people and these settlers is the Indigenous people got absolutely wiped out. So sure, there was warfare, there was deliberate armed genocide, there's no doubt about that, but a heck of a lot of the deaths took place because of infectious disease. So slot that in.
Sorry, go on, you go ahead. Sorry.
No. You take the baton.
Well. I just want to say that yes, that's perfectly true. That has happened at different times in the past, and large sections of the population just died because they were overwhelmed by the disease. They didn't have any preparation for it, and that would have been not just. They didn't have any genetic immunity to handle the overwhelming onslaught of the new disease. And would have that genetic immunity have occurred? Well, some of these processes I'm talking about, if they'd be given the chance to. But of course, it never
Yes.
happened that way. Uh,
Yes.
but
Yes.
so, but the so yes, it can, you can expand. That looks like, in that particular case, it looks like pure Darwinian selection, doesn't it? And it is in a sense, because it's the whole population is being affected. But let's just take another example from an animal population that was subjected to the same thing, and what happened at the edges of that. In the 1950s, up in early 50s, there were rabbit plagues all over Australia. CSIRO set up a program with myxomatosis to wipe out the rabbits, and it did. It wiped out 99% of the rabbits, but there was a small portion of rabbits that survived. And when you look at the sires, the male rabbits, this was the key thing that indicated we're dealing with some of the phenomena we've just been talking about. Sobey and Connolly at the CSIRO in Canberra, analysing all of that data from that period and the survivors, and actually did some experiments. They demonstrated the sire effect. And what was that? The sire effect was that a surviving myxomatosis male that had been, survived myxomatosis, if they mated that male, he got over the myxomatosis. If they mated that male to normal female rabbits, normal does, they could pass on something to the mother, who could show on cross fostering to other babies that she picked up something from the male that had survived. And it was one of these, suspiciously like these vesicles, these communication pathways I've just been talking about. So the sire effect, and then the population rebounded because they had some sort of immunity. And
Yes.
so there are real life examples of this in animal populations. And therefore, I would say that the survivors, if there were a few survivors of those, amongst the Mexicans or the Aztecs and these other people that really were affected dramatically, any of those survivors could have displayed the same phenomena. But of course, that was a long time ago in a different era when it's impossible for us to go back to that.
Yes. Yeah. And there just isn't.
All I want to say is, I agree with the points you're making, but that doesn't deny that Lamarckian processes go on, but it underlines why they would be important to genetically prepare a population for a disease.
Yes.
You see?
The other thing that occurs to me, I'm interested whether you think Lamarckian processes might be at play here, is let's take the example of smallpox, which when it was first introduced to Western populations, because I think its origins are actually in the East. They at least Persia and India seem to have had quite an ancient tradition of practices for handling it. But in any case, it was a very deadly disease. And then by the even by the late 18th century, and certainly by the 19th century, smallpox was widely seen as being a much more benign disease. And so I'm wondering whether each successive generation of parents, so people who actually had a smallpox infection and survived, were able to then pass on something to the immune system of their children, which made them better able to combat smallpox.
Well, yes, it's quite possible. That's all I can say. I'm not going to deny that. But look, but I've got to stress that I've been operating at the level of the basic processes that will allow these successful events to happen, and the evidence for them. I haven't been able to cover everything.
Sure. Yes.
Yeah,
I don't want to drag you into areas where you don't have detailed knowledge. I think,
I can give a general comment, but I wouldn't be very precise. Yes, go on.
No, absolutely no. I appreciate that. So what we've got so far is there was this division, largely an artificial division, in the history of the development of evolutionary theory between Darwinian selection processes and Lamarckian processes. That division probably shouldn't have even been there, given that Darwin himself was acknowledged truth.
It's an example, just like with COVID, huge political forces steered the whole direction of the events.
Yeah.
You
Well,
know,
once
just
that's a
personally, COVID wore me out because it took away 5 years of my life in a way to handle the vaccine controversy, the origin, and a whole bit. But there was just so much hysteria there. But just imagine there was a similar hysteria in the history of evolution. We've got to understand that similar hysteria. Yeah.
Oh, that's an incredibly important thing you just said. And I think I wasn't going to go here straight away, but hey, we might as well. This really underlines something about the nature of science, or more importantly, perhaps the nature of scientists, which is that contrary to this idea of that we non-scientists in the population are often given, that scientists float on clouds and all they're interested in is discovering the truth, and
Yeah.
they just really don't get up to any skullduggery like, destroying the reputations of their competitors, or falsifying their results or anything like that. That stuff happens a lot.
It does. Unfortunately, it happens a lot. The science is propagated by human beings with all the human failings, and
Yeah.
I've certainly been on the receiving end of a lot of it. I've tried to come through to the other end, try and be level-headed about it. I honestly have. I'm not trying to, necessarily blame, but it also explains why I've had to fight so hard as well at different stages. And therefore it affects you. To use a crude example at the political level, the Pauline Hanson phenomenon. But I'm not that at all. I'm far more, I, uh, I just wanted to, I've just been so interested in discovering, explaining the clear mechanisms that now exist for Lamarckian phenomena that can appear.
Yeah.
And
Yeah.
I
Yeah.
go on.
So in summary, we have evidence that that Lamarckian type processes of inheritance do occur, and we not me personally, but you and people you've worked with, and other peers
Yeah.
in this room, have elucidated the mechanisms by which Lamarckian inheritance could actually occur.
Yes. I'll give you an
They're the
example of the human, of the human portion. It's right at the tip, what's called the telomeric end of human chromosome 14, is a very large region, 1 megabase region, that encodes the antibody heavy chain. The point is that's a critical region. By that I mean it's the antibodies consist of heavy chains and light chains. There are the combining site of an antibody making it specific to neutralize that virus is a combination of a heavy and a light, but the heavy contributes a lot to it. Okay, but just to simplify things, that portion on chromosome 14, 1 megabase, that's 1 million bases, has been now sequenced quite a few times. And we're discovering, and this has been gradually growing on us for the last 25 years, we're discovering some extraordinary features about this, which only makes sense in that in every conception, there is a new somatic to germline infection event at that locus, changing the genetics. The whole locus looks like it's in a constant state of transgenerational churn. Now, that to me is predicted by all these processes that I've just gone through, the molecular, I only to briefly describe them, but the molecular process of entry of a new variable region element from a somatically mutated B cell, a lymphocyte, getting into the germline and then getting into that region, it will do things. It will scramble genes. It will create new things popping up in a place where they shouldn't have been. Like there was a standard gene in that place, but there's a new sequence related to it, but different at that place. And that's called an allele, by the way. Any variant of a gene, of a reference gene, is called an allele, but they're all genes. But the point is, you can see this, but there's also other, movement within the locus. So it's in a constant state of churn. Now, when you actually look at experiments that have been done in mice, of course, it's more pedestrian, it's more controlled and contrived. But when you look at humans, you're looking at the real world of what's going on genetically in this region. And this has led me to this following conclusion. At every conception, there is the opportunity in the immune system for a new somatic gene in this part of the chromosomes to enter the germline and replace or alter a resident gene. Now, that's an extraordinary position to reach, isn't it? When you think about it, because that says that every 1 megabase sequence that you see in human beings, in each individual, is different from the next individual. Now,
Mm.
that might not sound too, interesting unless you look at many other highly variable portions of the human genome, which have nothing like that extreme diversity. Because what that
Yeah.
says is that let's just say there are, well, I don't know, 7 billion people on the planet. Let's say in a diploid, that's 14 billion individuals. That means that it's, these are rough numbers in order of magnitude number. That means there are 14 billion individual sequences in that region. Now, you go to something like the transplantation system, a system that controls human transplantation, and self-identity from one person to the next. It's in a
Mm-hmm.
totally different order, of much lower down the chain. You can get repeat isolations of sequences, and you can, it's totally different order. And the point is, why is that? Well, I think it's because the immune system that is the tip of the spear of Lamarckian inheritance. It's going on all the time there, but it can, in principle, go on in other parts of the genome, like the olfactory. The olfactory genes, that are genes that encode the receptors in the nose that recognise all different volatile molecules, that we detect in our smell. That has a huge repertoire of germline genes, and they're spread around the genome. I won't go into the reasons for why they're spread, because even I don't know why they're spread around, but the point is they're spread around the genome. And
Mm.
that's an ideal setup for this latest technology. That's the last message of our paper, was that this technology that's being deployed to look at these really long regions of DNA could be deployed to other big clusters in the genome of olfactory genes, particularly in the experiment, which you wanted to probably talk about, but it hasn't come up, because it's really important. It gives an insight in how instincts arise. You know,
Yes. The olfactory
and
one, I
they're
do want
really
to
important. Yeah.
that. Before we go to that, I just want to tie a bow on the point that you've been making. So this 1 megabase region that has this incredible variability.
Yeah.
So what you're saying is that the point of this variability is that this is basically an opportunity for the immune system to produce maximum adaptation of that individual
But that
to their
family,
environment.
in that family tree,
Oh, yeah.
that individual, you see, we have to think of it in terms of that level. The emerging new baby appearing
Yep.
has got some of the experiences of the repertoire of antibodies necessary to survive in that, whatever part of the world that is, in whatever environment.
Yeah, yeah,
And
but this also means that okay, that the need of that region to adapt is going to be different depending on whether this is the firstborn child or a latter-born child who's got multiple siblings who are going off to daycare and playing with
yeah,
friends
yeah,
and home interesting stuff, and maybe there's pet cats and dogs and guinea pigs and birds and fish and God knows what
yes,
else.
exactly.
You
Yeah.
know, yeah,
We actually talk
yeah,
about they're the sort of things we should be looking at naturally now. Now there's technology to do this rapid sequencing. Well, it's not rapid actually; it's actually quite difficult to do. But the
mm,
technology is getting there. We could look at one of the benchmarks would be standard living three-generational families. We have the grandparents, the parents, and the children. And if you can get to the fourth generation, do the fourth, because I'm absolutely certain you would find some very interesting changes going on in these variable region genes of antibodies. There's no question about it in my mind. But of course, it hasn't
yeah.
been done. But it's in
Mm, mm.
theory now can be done in the light of all the technologies available. And so, by the way, I often get misunderstood because that's where I'm operating all the time. I'm operating from the point of view: is this possible or not from the current evidence we have? And
Yep.
if it is possible, then I open the door to it as a possibility, as a thing to look for in future experiments. So if there's a difference between physical theory and biological theory, it's really because biology is so diverse. Our predictions have to be of a different type. They're biological predictions rather than physical predictions, which is a rather different way, mathematically precise. Biological predictions are to do with processes and events and the likely probabilities of events occurring. That's how I've been operating. But look, can we talk about instinct? Because this is something that
Yes,
everyone can
yes.
identify with. When we look around us at the instincts we seem to have, like the falling sensation on a tree, or fear of snakes, or avoidance of certain foods, and instinctual. There's an instinctual thing, like the odor, the smell of a type of potential food will immediately mean that you can't, go near that, and you don't know why. But these are instinctual responses, and the human. There's a large number of instincts in different human populations, but there's fundamental ones that we all have. And when you think about it, how did instincts arise? Because instincts are very purposeful. It's not irrational to fear a snake or a spider, because there would have been experiences in our parental past that indicate that there would have been a learning process. Must avoid snakes and spiders. Look, I'm very trivialising a little bit, but because people do handle them. But the point is that's an instinct embedded in most people. And the snake handler has to get over that instinct to do the handling of the snake.
That's right.
But the point
It is possible to overcome it, but it takes some
it's
work,
possible to
doesn't it?
overcome. But the point is, where do instincts come from? And you see, they only make sense in a Lamarckian theory of evolution. Think about it, because they're highly purposeful and specific. How could that arise by chance? You see,
Yeah,
in
because
so many people.
the Darwinian explanation would be, okay, somebody has a mutation that makes them scared of snakes, and so they recoil from a snake and they survive, and their friend who didn't have the instinctive fear of snakes got bitten and died. And so therefore the person who's got the mutation that makes them afraid of snakes is more likely to reproduce.
But
it doesn't seem very efficient.
No, but it's also but it's deeper than that. It's a complex behavioural response on the part. So we're not doing, we're dealing with a complex behaviour. And so that comes back to the possibility now of doing these experiments. Now, the experiments were done about 10 years ago describing Pavlovian, that is conditioning experiments, where you associate one stimulus with another stimulus, and then you try and recall the original stimulus with just one of the triggers. Okay, the example I'm going to use is odorant conditioning, Pavlovian conditioning in mice, where they conditioned the parental generation to associating a particular chemical, say acetone or one of these other chemicals they used, where they knew what the germline gene was that could target that particular odorant molecule, and they coupled that with a fear response. They generated a fear response by every time the parent smelt this particular odor, it had a mechanism to give the mice a mild electric shock. Okay? So there was an avoidance response in the mice associated with that smell, and they showed all this. This was all specific. That's the important thing. All very specific for that odorant, and they could locate it in the genes. But when they did their breeding experiments, they did Pavlovian conditioning and then breeding experiments. They used the conventional through the male only down the generation. You see, so of course it was reversible. Now, my argument would be, given that the similarity of that with all the other things we've talked about, that if you did the interbreeding and inbreeding experiments, you could lock that instinct, that newly emergent instinct between that fear and that molecule into the germline. You see, that's how I think.
Just to be clear, the way the experiments were done were you take the male animal, and then he smells the smell, he gets a shock. He smells the smell, he gets a shock. Then he's bred with a female who hasn't been subjected to that, and then the offspring are exposed to the smell, and they show a fear response as if anticipating.
Yes, that's right. Yes, and that gets down to, again, the fifth or sixth generation if you follow that breeding strategy. So therefore, it falls in the category of soft Lamarckian inheritance. You see, and epigenetics. Yeah.
And there simply is no Darwinian process that could possibly explain that, is there? That's kind of the point. Like there's just
exactly. See, so
There's no
that's really touching on the emergence of instinct that is inherited,
yeah.
really inherited. So that's why I wanted to
Mm,
raise it. And of course, all of those things were all the rage in the 19th century in the criticisms against slow Darwinian evolution, as it was being propagated, not so much by Darwin, but well, he was in a way because he needed time, but
mm,
particularly the ones that you know were vehemently his supporters, they got extreme in the way they supported Darwin, and that's what created the divide between Lamarck and
mm.
Darwin. But anyway,
This included Francis Galton.
what's it? Yeah, I think so. Yeah.
Francis Galton. Yeah, I know. I know he was. Well, he was known as Darwin's bulldog, wasn't he? And again,
No, that
this
was
really.
someone else. The Darwin's bulldog
Oh,
was
sorry, that
someone
was
else.
Huxley. That was Huxley,
Yeah,
wasn't it?
Huxley. Yeah,
Yeah,
yeah. Huxley in particular was.
as Darwin's bulldog. Yeah,
Yes.
yeah.
Yeah.
Galton was his cousin, one of his many cousins.
Yeah. But anyway, look, the point was that the proponents, pointed to instinct as something that needed a proper explanation. Look very Lamarckian because it was highly purposeful
Yes.
behavioural changes going on, and
And incredibly complex. Yeah, yeah.
yeah,
So the other example that you gave in your paper was where either a male or a female animal is essentially rendered diabetic through chemical destruction
yeah.
of the
Oh,
pancreas.
that's really relevant. I mean, a big way to everything that's going on in the
Yeah.
world, as you would probably know. The problem with the obesity, incidence of type 2 diabetes, the whole metabolome problem is now
Well,
rampant.
I'll walk you through my thinking process on that one. So again, sorry for the audience. So the experiment is done this way: either the male or the female animal, prior to reproduction, has their insulin secreting cells destroyed by a chemical that's commonly used
Yeah,
against
yeah,
pregnancy.
yeah. That's a simple experiment. Yeah,
Um,
you take out.
then they're mated, and the offspring are essentially spontaneously diabetic. So they
Yes.
show a high blood glucose level. Now, in the case of the offspring of female mice, I was thinking, well, yeah, okay, so we know that in gestational diabetes, so when a woman who wasn't diabetic becomes diabetic during
Yeah.
pregnancy,
Mm.
because pregnancy is a state of normal
Yeah.
insulin resistance, but sometimes it goes off the scale and becomes abnormal. Well, her baby is more likely to develop type 2 diabetes down the line because it's been essentially bathed in an environment of high
Yeah.
glucose for its entire gestation. So that one I could kind of go, I don't know if you really need Lamarckian explanations for that one because we see this
No, no,
happening.
no. It's further down the line because they would say that, okay, that person's developed type 2, but the thinking is it's not genetic. Therefore, any type 2 person can marry another type 2 person and have children and not worry about it because it's not genetic. But the trouble is that's precisely the strategy that I would recommend in an experimental situation to consolidate the
Yeah.
change. And look, it's really extreme. It's really extreme in Australian Aborigines in remote areas. The incidence rates for type 2, then extending to kidney disease. It's just off the Richter scale, as you know, and it's a major political problem in Australia. But
Incredibly
I
tragic.
fear it's now been embedded in the genetics of these remote communities. That's my fear. That's what's happened, and the
You
only
think
way
it
we're
was
going to reverse it.
the same thing in the Pima Indians in New Mexico, where they just
Yeah,
have off the Richter scale rates of
yeah,
diabetes.
yeah, and
Their
go on.
genetic relatives, the Tarahumara, just over the Mexican border, have very low rates until they're
Yes,
introduced to Western diet. But anyway, so going back to the animal experiments, though, what I found far more difficult to explain via conventional means was the fact that or the development of diabetic offspring when it was the
yes.
father mouse or rat or whatever the heck that was rendered
Oh,
diabetic
yes, that's
through
the point. Is you
through
can't
this
just focus
chemical.
on the mother.
Um,
Yeah,
and so I was thinking, okay, well, how would I explain this if I wasn't using Lamarckian inheritance
yeah,
mechanisms?
yeah.
Well, okay, so we know that it's actually it's the father's genes that shape the development of the placenta,
Mhm.
and so the placenta plays this huge role in the development of gestational diabetes. But I was thinking, but hang on a minute, that would still require a Lamarckian process because and it's not even the father's genes that have been altered by this,
Yes,
right?
yes,
His genes haven't been altered at all. His pancreas was chemically destroyed. So,
yes.
yeah, you got me on that one. I couldn't come
No,
up with
no.
an
Well,
explanation
that's the
for
way
it.
those experiments were done. They were either done in the
Yeah,
male only or the female only,
yeah.
or both, and they were getting phenomena down the generations from either line. And those are well done. And of course, the stability in these lines, they're quite bizarre when you look at those the experiments. Well, they don't do those experiments anymore, but they're very informative because they really showed that you can get this. But look, there are other phenomena like acquired autoimmune disease, which there's only one clear experiment that was done. It was done 100 years ago. No one, people, it was quite famous back then, but it got buried by the hysteria around Lamarck at the turn of the 20th century. And that, unfortunately, but there, it was a really bizarre, highly stable Lamarckian transmission of induced eye defects. It's called the experiments of Geyer and Smith, where you target immune response against the eye lens proteins in the mother, or anyway, you get male offspring that have got this autoimmune disease phenomenon now, and they're propagating it to their offspring. And of
Mm,
course, it really escalates even further if you mate a partially blind male with a partially blind female. It just gets even worse. And they
gotcha.
ended up after 7 generations without totally blind rabbits.
Mm,
But we never do that experiment today. It wouldn't be allowed, I don't think, in ethics committees. But it was done then, but it indicates that as we'd expect, acquired autoimmunity, where the immune system now is being harnessed against its own tissue. It's in a
yes.
highly maladaptive, destructive way. So it's only when we see these extreme situations, of maladaptive behaviour that we can actually look at it and think, well, how do you explain these sort of things? Anyway,
Yeah, how
I've
can you
certainly
explain
done my
and that
utmost. Go sorry.
yeah. And that makes me think of Thomas Kuhn's notion of paradigm shifts, which is
Yeah,
a paradigm shift can only occur when there's such an accumulation
of
of
inconsistencies. Yeah,
yeah. So you've got this dominant paradigm and the paradigm predicts this, that, and the other thing. And then all
yeah,
these data are accumulating that don't fit that paradigm. They're
yes,
not what would be
that's
predicted
right. I
as
think
the
that's the main message from our talk today. There's a lot of data
mm,
that don't fit the paradigm. Yes,
yeah.
and I just thought, well, that's got to be at least highlighted again, because I didn't actually. The last time I tried to do this was 25 years ago as a semi-popular book. It really wasn't that popular in the sense that it could be understood easily, Lamarck's signature, but
Mm.
and I haven't really got into that yet. But look, I think that I think the take-home message is that the weight of evidence now favours Lamarck rather than Darwin. I think we've got
Mm.
to face that fact and try and be dispassionate in allocation of research funding and tolerance for people talking about these things. That might
On
be.
the notion of research funding for this, which I completely agree. I mean, it's just obvious that these mechanisms need further investigation.
Yeah.
What are the okay, given the economic model of funding, okay, we've got basic science research funding that's put up by governments and by some, I guess, not-for-profit institutions. But a heck of a lot of the research that's done in universities has some kind of commercial application.
Yes.
And so
Yes.
I'm
Yes.
wondering, what are the implications of Lamarckian evolution for funding for research, given that the funding model for research so far seems to me to have been very, very solidly grounded on the notion of evolutionary, of Darwinian evolutionary models.
Yes.
Sorry, did
Very.
I ask that question appropriately?
Yes. Yes. And uh, well, I think there are two things. There's the application of the knowledge, and there's the origin of these things. And I've always been
Mm,
concerned with the origin, the emergence of these phenomena, rather than how we apply them, which means that that's always put me in a position where I can't really write proper research grants. So I think we've got to try to get a balance back to funding basic research in its purest form,
Mm.
but and in a way that interacts with the applied and feeds and generates new ideas for the applied people, rather than just exclude the basic research, which doesn't appear to have any immediate application except understanding, actually understanding something. And I think that's a big difference going on now in the world between application and actually understanding something. Anyway,
Mm.
that's just my
Yeah,
views.
I remember I was in my final year, or final, maybe it was the second last year, I can't quite remember. But anyway, I was still in uni when the Human Genome Project was in
Yeah,
its
yeah,
sort of final stages. And I do remember it must have been early enough in the Human Genome Project where the expectation still was at that stage that the human genome was going to consist of somewhere around 120,000, yeah, 120,000 genes.
yes,
Because subsequently it turned out the human genome has about, what, 23,000, which apparently is less
yeah,
than a rice plant. That's quite entertaining. But again, when I was at uni and there was much chatter about the Human Genome Project, the thinking at that time was, we're going to sequence the human genome and we're going to unlock the keys to human disease, and we're going to have all of these fantastic treatments that are based on the fact that these diseases have this huge genetic component, and won't it be great, we can target these. And that didn't happen. But it does seem to be that research on the genetic basis or origin of disease is still kind of founded on that model.
yeah. It
And
is.
again,
It's on that model. Yeah,
yeah,
yeah.
and so again, how does a Lamarckian understanding, or does a Lamarckian understanding affect, have implications for
Well,
that?
yes. Well, in the sense that in all those steps that I put out now that clearly would have to be involved in any Lamarckian process, then all of those things have got to be then discussed ultimately in that context, not just in a pure Darwinian context of natural selection, but far more nuanced and complex a context, and
Yeah, it
and
just
the
does
origin.
seem to me. Yeah, I'm sorry. Go ahead.
Anyway, look, I really probably don't have much more to say now. I think many of the major points are, we've discussed, but it gives you a different perspective on the way things happen in genetics, so doesn't it? If
Yes.
genes.
I am hoping. I think I said this to you last time we spoke, that I have a couple of readers whose attitude toward genetics is, I don't believe in that stuff. I don't believe in DNA. I don't believe in. Well, some of them say I don't believe in evolution, or I don't believe in genetics, or whatever. And what I'm hoping is that, well, I hope those readers will actually watch this today. And
Yeah,
what I hope this will do is like, whatever you think you don't believe in or don't go along with, or whatever criticisms you have of the version of evolution
yeah,
that has been taught to you, I hope that this has given you some glimpse into a world beyond that
mm,
version that people, in my opinion, quite rightly have criticisms of.
yes, yes.
So I guess that's. Thank you so much for your time today. I've taken you well
Yeah,
past
yeah.
the
I think I
time that
probably
we had arranged.
expended everything at the moment that I can convey at the moment. But yes,
So final message, Ted, to the evolutionary skeptics, the evolution skeptics out there. What would you say to these people as a scientist who spent his entire professional career in this
well,
domain?
the first message is that, of course, evolution occurs, but the mechanism by which it occurs is not the simple, slow, and ponderous process, by conventional Darwinian theory. There are more influences coming in and speeding it up and directing it. That's all. But
Yeah,
the
terrific.
idea of binding in the dark or natural selection as a principle, of course, I'm not going to deny that. But when you have feedback loops and other things going on and transgenerational effects, there are other influences now. impacting on human evolution and animal evolution, both of us. We're all in the same boat in that way. Yeah.
Yeah. Yeah, terrific. All right, thanks. It's been such a treat, Ted. And once again, thank you so much for being generous with your time. I'm so glad you sent me that paper, and I will, of course, include links to that paper and anything else that you want to include, including maybe your book, Lamarck's signature. I'll put all those links in the show notes for people who want to follow up on this.
The other note I sent you, of course, is not published. It's still under review. They haven't rejected it. But the point is, so that's but the ones that are published, yes, the ones that are clearly published, yes, of course.
Tremendous. All right, we'll do that.
Good,
okay, and I hope to speak again soon, Ted. All the best. Thank you so much.
Thank you very much, Robin.